Welcome once again, Neuronovas, to the second part of our first series, which is living things. Now we are going to be talking about cell structures and functions. Are you there now? And the first component we want to discuss is the nucleus. But before we start the concept, please do us a favor of subscribing to our channel and hitting the notification, as well as commenting below wherever you have a question. Okay, good. Now, you have the structure on the board, and the structure on the board is not the cell. This is a nucleus. I've isolated the nucleus so that we can really see all the components of the nucleus. Yes, this is the reason why so many questions that are presented to you are being misinterpreted, and then you tend to pick wrong answers. Now, you need to understand individual organelles properly. You understand? JAMB is not just about you knowing that, the nucleus is responsible for cell division, or it is the most important part of the cell, or all those kind of, no. You're writing an exam that tests your ability to identify misconceptions. Okay, now, in this diagram here now, which is the nucleus like I told you, first thing I want you to see here is that I used double line to show the boundaries of the nucleus. Now imagine every other portion here is the cytoplasm of the cell. I get me now? Just imagine every other portion here is the cytoplasm of the cell, and this is the nucleus. That means this structure here separates the nucleus from the cytoplasm, right? Good. So it means that everything within these compartments here, that's inside the nucleus, everything inside the nucleus here is called the nucleoplasm. It's called the nucleoplasm. I'm going to be adding a structure here, very important. I have to add this structure here. This is a very important structure. We will still discuss that. So it's called the nucleoplasm. Every other thing inside here is the nucleoplasm. Now, what are these double lines? What are these double bound structures? What are these double lines here representing? They're representing the nuclear envelope. The nuclear envelope. So I'll just do that. I'll do it this way. I'm going to bring it in this direction. So these two things here represent the nuclear envelope. The nuclear envelope. So one of the things we can identify there about the nuclear envelope is that what is a double membrane bound organelle. So the nuclear envelope is a double membrane bound organelle. You can see that it is made up of two membranes. Each line is representing one membrane, so it's double membrane bound. So a question can come up to you and they'll just ask you, which of the following is a double membrane bound organelle? And you see nucleus, that's it. You don't need to go further. Once you see nucleus, that's it. We have other double membrane bound organelles, but for nucleus to be the option, then that's it first. I guess now, before we wrap up the lectures, I'll give you other organelles that are double membrane bound. Okay, so you have the nuclear envelope, and then we also have these structures that are here. You can see them here, all these red structures here. I get me now, and if you look at them carefully, they look like gateways, right? So common sense. And then we'll call it the nucleopore, or you call it the nuclear pore. So we have the nuclear envelope, which is double membrane. So when we say it's double membrane, it means it has a what? It has an inner membrane. An inner membrane. We can call this line that is more inward. We can call it the inner membrane. And we also have the ones that are the outer membrane. They do not perform exactly the same function. Yes, they do not perform exactly the same function. That's why you must understand that here, that nuclear envelope are double membrane bound. Each membrane has specific functions that they perform, aside from the general membrane functions. You understand? They have their unique functions. All right. So here we have nuclear pore. Nuclear pore. I get it now. So nuclear pore clearly is just for transport of materials in and out of the nucleus. This is the nucleus. So materials want to come from the inside of the nucleus out, and some want to go from outside the nucleus in. Like, to make it more specific, I'm trying to say that some materials want to move from the where? They want to move from the cytoplasm into the nucleoplasm, while some want to move from the nucleoplasm into the what? Into the cytoplasm, that's out of the nucleus. Alright, so that's one structure we could see there. Another structures we can see are these structures here. these structures that are here like this, these structures, including these ones here, they are all called what? Chromatin. They are all called chromatin. Chromatin. You see, I just need to get something. Chromatin are the non-condensed forms of your chromosome. Yeah, when cell division is about to start, the chromatin will condense. And when they say condense, the condensation of chromatin or material simply means that they grow shorter and thicker. This has come out in JAMB before, multiple times. So when you hear the word chromatin are the non-condensed form. So when chromatin gets shorter and thicker, they become chromosomes. That means they've condensed. So when they condense by growing shorter and thicker, and that will make it a chromosome. Okay, so these are the chromatins now. That's like saying the chromosomes, just that they are not yet condensed. Okay, good. Now, these are the chromatins. We want to know what are the specific functions of each of these things we just labeled now. Yeah, I've not labeled this guy here. This red guy here is called the nucleolus. The nucleolus. Now, these names, I'm sure that some of them are, you are familiar with some of these names, but I need you to understand the functions of these things. As neonova scientists, you need to be very, very detailed and very in-depth with the concepts that you are presented with for examinations. That's what assures you to be able to hit top scores in those courses. Now we've expatiated on the materials that you find within the nucleus. So in case they ask you what are the materials you find within the nucleus has a nuclear envelope. You can see that there, and the nuclear envelope is double membrane bound. That is double membrane bound. It has two parts. I get now, which is the inner membrane and the outer membrane. Let's start from that inner membrane. The inner membrane of the nuclear envelope, which is this side here, you see that there are these green things that lines the inner membrane. They are lined by these green things here. These green things here are called lamins. They are special proteins that are called lamins. And then also the inner membrane is lined by structures that we call lamin. Yes. And this lamin, what do they do? Now, I want you to notice some of these chromatin. There are some chromatins that are sitting directly on top of the lamins. I see now, you can see this chromosome here, chromatin here is sitting on this lamin. This one here is sitting on the lamin. Now, the lamin helps to hold tight chromosomes. Are you there now to the inner membrane? So they can help to hold firm. They help to hold firm chromatins. They help to hold firm chromatins. Another thing lamin can do is that they also take part in cell division. They also aid in cell division. So you can see when they just generally tell you that, oh, nucleus aids cell division. Either now nucleus aids cell division. You can see that one of the components in the nucleus that is aiding that cell division is the lamin. In fact, there is this condition that arises from mutation in lamin that we call progeria. Some people call it the Hutchinson-Clifford syndrome. Yes, it's still the same. So it's a condition that can arise as a result of what? Mutation to this lamin, precisely lamin A. But as JAMB, I just need to know, okay, the inner walls of the what? Of the nucleus, the inner membrane of the nucleus are lined by a protein called lamin. And what do lamins do? They hold firm the chromatins, which you see here, and they can also help in cell division. If they ask you mutation to the inner mutation to which structure or which component of the inner membrane can lead to progeria, you know that it's lamin. Okay, so good. Now, how about the outer membrane? Now, the outer membrane of the nuclear envelope, is usually rich in ribosomes. You see them there. You see ribosomes there lining the outer membranes. So why the inner membranes are rich in lamin, the outer membranes are rich in ribosomes. They are rich in ribosomes. Do you get that now? So let's recapitulate. Nuclear envelope divided into two. The nuclear envelope, that is the nuclear membrane, is a double membrane bound organelle or structure, and then is made up of inner membrane and outer membrane. The inner membrane is rich in lamine A, while the outer membrane is rich in what? Ribosome. Lamine A hold firm chromatin and aid in cell division, while outer membrane contain ribosome. Ribosome generally does what? Protein synthesis. They help you to synthesize protein. So protein synthesis. Protein synthesis. So that's what it does. That's what it does. It helps you to synthesize protein. Now, how about the nuclear pore? Basically, the nuclear pore just aids in what? Transport. It helps in allowing transport of materials both in and out of the nucleus. So materials can enter into the nucleus and materials can go out of the nucleus. So that means that there are some materials that need to leave the nucleus, and there are some materials that need to come into the nucleus. Example of material, because JAMB has asked that question before. Which of the following materials are transported out of the nucleus? Which of the following materials are transported in to the nucleus? So one of the materials that are transported in, transported in, because we said that materials can go in, and we said that materials can go out, can go in and out. So some of the materials that can go in, you have the histone. Are we there now? We have the histone protein. It's a protein that we need within the intracellular compartment of the nucleus. Yes, we need it in the nucleoplasm. This is histone, and I will show you why. It's a major requirement when it comes to the formation of chromatin. That's it. We need it for the formation of chromatin. Chromatin is usually DNA plus histone. You understand? So you have DNA, you have histone, you form the chromatin. You see that now? So that's basically, so this histone is usually generated from what? From the extracellular compartment, that's like the cytoplasmic compartment. That is all the compartments outside the nucleus. So it's then transported into it. It's generated from that your common amino acid that we call histidine. So that's where you get this histone from. Is a protein. Okay, good. Example of materials that usually want to go out. Example of materials that usually want to go out is what? Ribosome. Ribosome want to go out. Are you there now? And RNAs. RNAs also want to leave. Because we have RNA within the nucleus, and you also have them outside of the nucleus. So those are examples of stuff that usually want to either leave or stay within the nucleus or transported in and out. Okay, good. Now we've talked about the nuclear pore. How about we talk about the nucleolus? What's the function of the nucleolus? Now the nucleolus, basically, if you read textbooks or materials, they just go straight to the point, and then JAMB does not know, okay, what kind of material you read or what your foundational knowledge about the concept is. All they just need is answer their question. So you need to be very knowledgeable. So some textbook just tells you that nucleolus houses ribosome, right? Yes, not a lie. But basically, I'll put to you that you need to understand that the nucleolus actually contains rRNA, which is the ribosomal RNA. It contains the ribosomal RNA, which is a major component necessary for the synthesis of the ribosome that you're already familiar with, that is stored within the nucleolus. So they just tell you, oh, nucleolus houses the ribosome. You need to understand how the ribosome gets to the nucleolus, or how the nucleolus produce or synthesize the ribosome. Good. So first thing first is that in the nucleolus there is usually rRNA. Are you there now? And this rRNA tends to then combine, are you there now, with a protein that we call ribosomal protein. Ribosomal protein. So the combination of rRNA with ribosomal protein is what leads to the formation of what? It's what leads to the formation of ribosome. It's what leads to the formation of ribosome, or you can say the synthesis of ribosome. So basically, what I need you to take home from the nucleolus there is that nucleolus helps in the synthesis of ribosome, or you can say it also houses the ribosome. Normally, when the ribosome has been produced, like we said, ribosome is an example of material that want to leave. So when they've been produced by the nucleolus, they move out and find themselves attached to the what? Attached to the outer membrane. That's why we said in the outer membrane you find ribosome. So when it has been produced from the nucleolus by combination of rRNA and the ribosomal protein, it then exits through the nuclear pore, are you there now, to come and attach themselves to the outer membranes. When they get to the outer membrane, some cascade of operations will happen, which will then cause the release of those ribosomes towards the reticulums, and I'll explain that in our next lecture. So good. So we've discussed nucleolus. Finally, for the nucleus, we'll be discussing chromatin. Now, I need you to look at something. In this diagram, we have some chromatin that are suspended, like more at the middle. They are not attached to the sides. They are not attached to the walls. You can see them here. Are you getting me now? And we have those that are firmly attached to the inner membranes, right? Good. So those chromatin that are more suspended at the middle, that are not attached to the walls, we call them euchromatin. Euchromatin. So we are trying to say now that chromatins are of two types. And what are they? We have the euchromatin, and we have the what? And we have the heterochromatin. Are we following now? Now, the point there is euchromatin structurally are found at the middle of the nucleus, but heterochromatin are found more attached to the inner wall via what they call it. I mean, if you remember, I said it holds firm the chromatin. So now you know the kind of chromatin we are talking about, that they hold firm the heterochromatin. So basically, what the general makeup of the chromatin? The chromatin, like I said earlier, is just what? Is just DNA plus what? Plus histone protein. So that's chromatin. Chromatin is just DNA plus histone protein. Now, the major difference between the euchromatin, aside from what we said earlier, and the heterochromatin is that what? Is that the euchromatin have what we call loosed. You know, they both have DNA plus histone, because that is what chromatin are made up of. But the euchromatin have loosed DNA. Loosed DNA. Their DNA are loosed. While heterochromatin has tight DNA. So in the euchromatin, the DNA strands are loosed. In the heterochromatin, the DNA strands are tight. You understand that now? So that's basically it. So before we wrap up the lecture now, we can see now that DNA can help. Nucleus can help in what? In cell division, right? And also they can help in a certain number of processes. And what are those processes? Look at this. One of the things the chromatin will do is that they can turn DNA to DNA. They can help you convert DNA to DNA, or they turn DNA to RNA. Yes, we'll get and we'll explain the mechanism of these conversions in later lectures when we get to those parts in the JAMB syllabus, because they are there. Now, when the chromatin convert DNA and use it to produce or form or synthesize another DNA, we call that thing DNA replication. DNA replication. We call that DNA replication. But when DNA are used to produce RNA, you call it transcription. And now I know you're conversant with that word, transcription. So you have transcription. So we have transcription, and we also have DNA replication. So basically, the nucleus will help in cell division, the nucleus will help in transcription, forming RNA from DNAs. Then you also help you to form DNAs from DNA via DNA replication. You see that now? So that's basically it about the DNA as a JAMB bite. So try to keep these terminologies in your head, revise them, go through those things. If you have questions, drop them in the comment section and we'll respond to you duly. So for now, that will be that for nucleus. Are you there now? That will be that for nucleus. Just to recapitulate, remember the nucleolus synthesizes ribosome by the combination of rRNA and ribosomal protein, right? Also remember that your nuclear membrane is a double membrane bound structure where you have inner and outer. In the inner, you have lamins, right? of which mutation to those things will cause progeria. We have what they call outer membrane, which is where you find ribosomes attached to them. And then we talked about chromatins, the loosely packed chromatins, our euchromatin, the tightly packed are the heterochromatins. Chromatin generally is DNA and what? And histone. Very important. Take note of that. Then we also made mention of the fact that your chromosomes are simply the condensed form of chromatins. So during cell division, the chromatins get organized to become a chromosome. So that'll be that for that, Neuronova scientist. We'll be expecting you in our next lecture.